Bismillahirrahmanirrahim. Today the topic of my discussion is electron transport chain, that is also called as biological oxidation. The steps that are involved in electron transport chain are oxidation, reduction, and we will discuss about redox pair, what is redox potential, electron transport chain and various sites and complexes associated with it, and how the electron is transferred from FADH and NADH to the final acceptor oxygen. The basic thing which you have to remember is that the electron is transferred from the whole process and the final acceptor of electron is oxygen. Oxygen accepts that electron and the final acceptor is oxygen, that is the main point. Simultaneously there is the transfer of hydrogen in intermembranous space. We will also discuss how hydrogen is transferred, how hydrogen is pumped across the membranes. Then is the chemiosmotic theory, oxidative phosphorylation inhibitors, uncouplers. These are also few topics associated with the electron transport chain in next few lectures, but today the main topic of our discussion is the main electron transport chain and what happens in it, what are the complexes associated with it. We will discuss each and everything in detail. First of all, we will discuss some things that carbohydrates, proteins and lipids are the main constituents of our body, main organic compounds like sugar, proteins, lipids, and carbohydrates are converted into monosaccharide units and proteins are converted into the amino acid. Lipids are basically converted into fatty acid and glycerol, but basically fatty acids are transported to the adipose cells, and these are the monosaccharide units and amino acid units and fatty acid units. These are the simpler units that are called monomers, which combine to form the complex unit carbohydrates. And some carbohydrates we intake from our foodstuffs, and the proteins and the lipids that we intake from our foodstuffs are also converted into the simpler units in our body. These substances then enter into the TCA cycle and release carbon dioxide. And this is not the rule basically always that the products always enter the TCA cycle. Some products also enter the glycolytic pathway, as we shall see later in the topic of lipid metabolism, that how glycolysis occurs in the adipose cells. This is the second stage of metabolism and also known as the intermediary role of metabolism. What is the second stage? First stage is the polysaccharides are converted into the monosaccharide. The polymers means the large compounds are converted into the small compounds, and the small compound then enter into the TCA cycle or glycolysis for the production of energy, that is the ATP. Simultaneously there is a formation of NADH plus H positive and FADH. These two compounds enter into the electron transport chain, and this is known as cellular respiration. What is cellular respiration? When NADH and FADH form and they enter into the electron transport chain. Obviously when they enter into the electron transport chain, they produce energy and they start the process for the transport of electron to the final acceptor oxygen, and that's why this is known as cellular respiration. Oxidation will happen here, reduction will happen here. We will study about the redox pair, redox potential. What is redox pair? This is the redox pair. What is the redox? First of all, we have to learn about what is redox. Redox reactions are those in which the oxidation and reduction occurs simultaneously. And what is oxidation? Oxidation is the loss of electron basically. If we discuss it in terms of electrons, then the loss of electron is known as oxidation and the gain of electron is known as reduction. See, when iron in the form of ferrous Fe2 positive loses electron, then its positive charge increases, because when it was in zero state, then 1 electron lost, +1, 2 electrons lost, +2. Obviously when 3 electrons are lost, then +3 charge came on ferrous, and it becomes ferric. Fe2 positive loses 1 electron and it becomes Fe3 positive, and this is oxidation because it is the loss of electron. Then Fe3 positive if gain electron again, then it becomes Fe2 positive, and this is the reduction reaction. So this is the redox pair. Oxidation and reduction is occurring simultaneously. Similarly, some few examples are there which are the example of redox pair, that is NAD and NADH and NADH to NAD, FMN to FMNH2 and FMNH2 to FMN. Okay, there is also one more thing, that oxidation is not only the loss of electron, and we know that loss of electron, loss of hydrogen, these are the oxidation processes, and also the gain of oxygen is also oxidation, and gain of hydrogen, gain of electron and loss of oxygen, totally inverse of the oxidation is the reduction. So if the gain of electron occurs, if the gain of hydrogen occurs, then it is a reduction process. So these are the oxidation reduction pairs, that are the redox pair, NAD and NADH, FMN and FMNH2 and FAD and FADH2. These are the redox pairs that are present in our this discussion. Now we will discuss about some electron, as we have seen that this is the oxidation, reduction, then redox pair. We have discussed about the redox pair, that what is the redox pair. Now we will discuss something about the redox potential. What is the redox potential? This is the redox potential, like this. Redox potential of some processes are given there. Here redox potential, electron transfer potential, it is also called as electron transfer potential. NAD and NADF2 minus, some have the minus redox potential and some have the redox potential in their positive values. And when we will now discuss the detailed description of electron transport chain, then we will discuss that why these are positive and negative values. For few compounds there are positive values of redox potential, and for few compounds there is the negative value of redox potential. So this, what is this? These are positive value, negative value. Now when we will discuss the full process of electron transport chain, we will you will learn most of the things that are related to the electron transport chain. Now see the site of electron transport. This is very important thing regarding MCQs, regarding your concepts. The site of electron transport chain is inner mitochondrial membrane. This is the inner, not the outer. Inner mitochondrial membrane. Mitochondria have two membrane, mitochondrial envelope, and are consist of outer membrane and the inner membrane. The inner membrane, inner mitochondrial membrane is the site where the electron transport chain occurs. There are four main complexes in electron transport chain, and the fifth one is ATP synthase, which is responsible for the synthesis of ATP. That means there are four complexes in the membrane, and the fifth one is the ATP synthase, that is responsible for the transport of hydrogen ions. Now this is the diagram related, this is the mitochondrial structure, and you can get a lot of mitochondrial images from Google. Go to the google.com and search for the mitochondrial structure. You will get a lot of structures of mitochondria, and you have been studying from school level the structure of cell and the structure of mitochondria. This is the mitochondria, outer membrane and the inner membrane, and intermembranous space, the space between the two membranes, and below this there is a rough sketch of how electrons are transferred. Now we will discuss it thoroughly. This one, electron transport chain, complex 1, complex 2, then there is the complex 3, and then is the complex 4. There are the four complexes, as we have told you, as I have told you earlier. This one, there are four complexes, and fifth one is the ATP synthase. Complex 1 is NADH coenzyme Q and oxidoreductase enzyme, and along with it FMN and iron sulfur protein is associated with it. Here NADH plus H positive From here electrons are transported to FMN. FMN is converted into the FMNH2, and then through the iron silver protein the electrons are transported to the coenzyme Q. Then the complex 2 is the succinate, then coenzyme Q, succinate, coenzyme Q, and reductase. Associated with these are FAD and iron sulfur protein. From succinate electrons are transferred to FAD, and then through the iron sulfur proteins these are transferred to the coenzyme Q. That is the complex 3. First of all, see the diagram. I will show you a diagram here. This one. These are the 4 complexes. As you can see, the 4 blocks you are watching. These are the 4 blocks above which I have written the hydrogens. These are the 4 blocks. These are the 4 blocks. As you can see, 3 large boxes and 1 small box in the membrane. This is the membranous structure, and there are the 4 blocks in it. These 4 blocks are the 4 complexes. For the rough sketch, that these are the these are the 4 complexes that are embedded in the membrane. Now we are watching the complexes, that what are their composition and how electrons are transferred in these complexes. Then is the complex 3, coenzyme Q and cyclic cytochrome C, coenzyme Q, cytochrome C oxidoreductase, and then along with it associated is the cytochrome B, cytochrome C, and iron Fe2 positive, Fe3 positive. Next is the complex 4. In the complex 4 there is cytochrome C, which is not written clearly in the slide. This is a cytochrome C, and then is the oxidase, and along with it associated is the iron Fe3 positive, as it is Fe3 positive, Fe2 positive, then is the Cu2 positive, Cu positive, and the electrons are transferred from cytochrome to cytochrome A3, and then electrons are finally transported to the oxygen, and then this oxygen is used to make water in the body. So we have seen there are 4 complexes. Electrons are transferred from the first complex to the third and then fourth, or electrons are transferred from the complex 2 to the further steps. So the first complex, as we have seen how electrons are transported through second complex, how electrons are transported from which compounds, and from third complex how electrons are transported, and from fourth complex how electrons are transported. Coenzyme Q is also called as ubiquinone lipid with isoprenoid unit is known as ubiquinone. So this is also known as ubiquinone. It is a mobile electron carrier. Its position is not fixed. Coenzyme position is not fixed in the cell. Cytochrome C is also an electron carrier. Now there is a diagram of electron transport chain, and 4 large blocks are 4 complexes that I have discussed earlier. The 4 complexes are fixed in the inner mitochondrial membrane. These are fixed in the inner mitochondrial membrane. Complex 1 NADH and coenzyme Q oxidoreductase, and this is only the name of the first complex that which compounds are present in it. Substrate for NADH, glyceraldehyde 3-phosphate, pyruvate, isocitrate, alpha-ketoglutarate, malate. These are all the compounds that enter into the glycolytic pathway or into the TCA cycle for the generation of NADH. In the TCA cycle, isocitrate, alpha-ketoglutarate, malate all enter into the TCA cycle and produce NADH, and in the glyceraldehyde 3-phosphate that completes the process of glycolysis and produce NADH, and similarly pyruvate also enters the TCA cycle and it also produces the NADH. So these are the substrates for NADH that produce NADH. Then the NADH produced within the cell undergoes this process that we can see in the slide. we can see on your screen that NADH enters the first complex in the inner mitochondrial membrane. Then there is FMN converted into FMNH2. The electrons are converted transported to FMNH2. Then FMNH2, the electron are transported to iron sulfur protein. Then electrons are transferred to coenzyme Q, then cytochrome B, then cytochrome C1 to cytochrome C, and then to the cytochrome AA3, along with are associated the ferrous and copper, Fe2 positive and Cu2 positive, and then electrons are transported to the oxygen, and finally the water is formed. How FADH2 is produced in the cell? Succinate fatty acyl CoA, mitochondrial glycerol 3 phosphate. These are the mitochondrial glycerol 3 phosphate. Basically it is glycerol 3 phosphate that is present in the mitochondria, which is responsible for the production of FADH2. And where the glycerol 3 phosphate is synthesized, glycerol 3 phosphate is synthesized mainly in the oxidation of fatty acid. In the fatty acid metabolism, glycerol 3 phosphate is produced, and that is responsible for the when fatty acids break down basically in the beta oxidation. Then they in the breakdown of fatty acids, basically if we say that the breakdown of a lipid, then lipid form fatty acid glycerol, then the fatty acid again break down to form energy in the cell, then they produce FADH2. Now we can see the first step, that in the first process of electron transport chain, the complex 1, complex 3 and complex 4 are involved for the production of water molecule. And the second process is that the complex 1 is not involved, but the complex 2 and then 3 and 4 are involved. Complex 2 is succinate, then FADH, FADH2 to FES, that means the electrons are transported from succinate to FADH, FAD and FAD is converted to FADH2, the electrons are transported to FES, iron sulfur protein to coenzyme Q, then FES is then again iron sulfur protein, then cytochrome B to cytochrome C1, cytochrome C, cytochrome AA3, again the whole process is repeated. But now here one more thing that you have to learn is, as we can see in the previous slides on the complex 1, complex 3, complex 4, I have not written anything about the hydrogen ions, but you can see that on complex 1, complex 3 and complex 4 you can see hydrogen ions. On above complex 1 there are 4 hydrogen ions, above complex 3 there are 4 hydrogen ions, and above complex 4 there are 2 hydrogen ions. Basically when electrons are transported along these complexes, these electrons while passing through the complexes produce these steps produce a lot of energy. That energy is responsible for the transport of hydrogen ions along the membrane. The hydrogen ions are transferred across the membrane, they are pumped out of the membrane. When the hydrogen ions are pumped out of the membrane, then the membrane is itself impermeable to the hydrogen ion. It does not allow the hydrogen ions to enter into the mitochondrial membrane. As the hydrogen ions cannot enter the mitochondrial membrane again, so the membrane is impermeable to the hydrogen ion. So the fifth ATP complex, ATP synthase complex, that is a complex of an enzyme. So the fifth thing is, the fifth complex ATP synthase then enters is responsible for the transfer of hydrogen ions from the inner mitochondrial membrane. So we can see that the hydrogen ions are pumped out of the membrane, and these cannot enter into the mitochondrial membrane again. Now the question arises that why there are no hydrogen ions pumped out of the second mitochondrial complex. The hydrogen ions have not transferred from the second mitochondrial complex because the second mitochondrial complex do not produce enough energy to pump the hydrogen ions. The energy produced here is very low. That's why no hydrogen ions are transferred out of here. In the first complex, second, third complex and fourth complex, there is a lot of energy, about 12 to 10 kilocalorie. So that's why hydrogen ions are pumped out of these complexes. Energy is released, 12 kilocal and 4 hydrogen ions are pumped out of the membrane. In the second complex, the energy is less generated and it is not enough to pump out electrons. As I have told you earlier, that in the second complex, the energy is less generated and it is not enough to pump out electrons. In the third complex, also energy is released and 4 hydrogen ions are pumped out of the membranes, and fourth complex, 4 hydrogen ions are produced. Membrane gradient is produced. Gradient means potential is produced and more hydrogen ions out of the membrane than the inside. So inside is negative, more negative, and mitochondrial membrane is impermeable to hydrogen, so it does not allow the entry of hydrogen. So the only source is the fifth complex, that is the ATP synthase. After the 4 complexes, you can draw another complex, like another box, that is the ATP synthase. My next lecture is totally about the structure of ATP synthase. You can listen to that lecture, the ATP synthase, what is ATP synthase and how it allows the transfer of hydrogen ions from the across the membrane, that is the chemiosmotic theory. In the next lecture you will learn more things about this. That's why it is chemiosmotic theory is related to the electron transport chain, because in electron transport chain, electron is transported in a whole chain and hydrogen ions are pumped across the membrane, and then to allow the entry of hydrogen ion, there is another complex that is known as ATP synthase, and next topic begins, that is the chemiosmotic theory, that is associated with the electron transport chain. That would be discussed in the next lecture. Listen to the next lecture carefully. Hope you have understood. If you have any question, you can ask in the comment box. Thank you very much.